Hello, everyone, and welcome to today's webinar. Spatial Transcriptomics reveals insights into the melanoma tumor microenvironment. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Illumina and 10x Genomics. To learn more about our sponsors, please visit www.illumina.com and www.10xgenomics.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speakers, Dr. Aida, Dr. Stephen Hague, Kim Thrane, and Dr. Hanna Eriksson. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Our speakers will now begin their presentations.
Hello. Today, I would like to talk to you about exploring biological systems at single-cell resolution with Illumina sequencing technology. Let's start by jumping right into the science. This is an article from the single-cell space that has been attracting quite a bit of attention. It is a joint effort between the Broad and the Massachusetts General Hospital. And what these researchers did was to profile around 7,500 cells from the windpipe. The nice thing is that they did it both using the 10x approach and a FACS-based sorting full-length RNA1 as well. Afterwards, all the sequencing was carried out using the NextSeq 500 system. When they analyzed the data, they saw that the windpipe's lining consists of 7 different cell types and not of 6 different cell types, as was previously believed. The interesting finding here is that this new cell type, the ionocyte, depicted here in purple, expresses a high level of the cystic fibrosis transmembrane conductance regulator, or CFTR, which is basically the protein responsible for cystic fibrosis. This means that the current understanding of the cellular basis of cystic fibrosis is likely to be incomplete, since instead of being a common cell type expressing CFTR being involved in cystic fibrosis, it might be predominantly this new, unknown until now, cell type that is involved in this disease. This is something that wouldn't be evident from bulk studies, because then all the cell types would just have averaged out each other, and you wouldn't be able to find these rare but very critical 7 types of lung cell. Well, this is just an example from the literature, but of course, there are many more. Taking a look at the trend in single-cell publication numbers, you can see here that it's almost an exponential growth curve. The reason why the graph here starts in 2009 is because in that year, the first single-cell sequencing article featuring NGS was published. And as you can see, there has been a 40 times increase in the number of publications since then. Taking a closer look at the 2018 single-cell sequencing publications in high-impact journals, we will see that there were 20 in Nature, 15 published in Science, and 25 in Cell, adding up to a total of 60 articles, and that meaning that there was more than one published per week. So as you can see, single cell is increasingly being applied, and it's impacting multiple areas, like cancer research, immunology, stem cells, and development, as some examples. So why single cells? Well, because each cell is unique, and having the ability to look into individual cells can tell you more about the underlying biology. In this example that we see here, we have two collections of cells, A and B. If you treat them both in a bulk fashion and you look at the blue transcript, you will arrive at the conclusion that the total transcript number in population A is 6, and that the total transcript number in case B is 6 as well. So this may lead you to the conclusion that these two situations are exactly the same, but these are in fact two very different biologies. In case A, all cells express the blue transcript to a certain extent, whereas in case B, most of the cells are silent, being only a few cells that express it to a very high level. So having single cell resolution, you can actually tease case A and case B apart, and that can show you more about the underlying biology. Like would be the example that we just saw in the cystic fibrosis paper, where instead of the cystic fibrosis receptor being in a common cell type, it may be more confined to a rare cell type that expresses it at higher levels. And this may lead to a very different conclusion. What we're also seeing is that there are many different new methods coming out in the single cell world. Initially, single cells started mainly in the RNA world, and right now you can profile 3', 5' RNA, full-length RNA, and you can also look at doing targeted RNA. But currently, there are also many other biological molecules that you can look as well, like for example, proteins, targeted DNA, and you can also look at different regulatory features, like for example, chromatin accessibility. There's also a very important trend towards multi-omics to see as much as possible or to get as much information as possible from these single cells. One example of this being the fact that site-seq approach is becoming quite popular since it allows you to look at both RNA expression and protein expression in the same cell. So as you can see, single cell is no longer a niche application, but it's being more and more broadly and more routinely used. There are also many technological advances being made and developed, and with these technological advances, cells can be isolated faster, easier, and more in a streamlined mode, as well as being able to study all these different types of biological molecules that we just mentioned, and not only RNA. There are also many improvements and developments happening in the analysis side of things. These include graphical tools, graphical interfaces to help you analyze single cell data in an easier way. But there are also groups working on getting more information out of the existing data. One example being shown here, and it's RNA velocity. It is basically an ingenious bioinformatics tool that tells you where the cell is actually heading based on its RNA data. From an RNA-seq experiment, you can get not only where the cell is based on its RNA expression, but also where it's heading. In this example, you can see, for example, the gray cell in the gray group that's in the right upper corner. You can see its vector, its RNA velocity pointing toward the direction of the yellow group of cells. So it's most likely that this cell is going to become this yellow cell type. With this, what you can do is follow cellular trajectories and see how development is proceeding. The beauty of this application is that you do not need to rerun your experiment, actually. You simply need to reanalyze the data that you already have. So all this basically shows that single cell is not a one-size-fits-all application. You can have different study aims, or you can look at different molecules, or you can do it on different scales. But one constant is that we at Illumina support all single cell applications with our wide range of systems. No matter what you want to do, we have a sequencing system that matches your output needs. From the iSeq, which could be used for library QC, all the way up to the NovaSeq, which will enable you to do large-scale single-cell studies featuring hundreds of thousands of cells. So if you have any questions about your particular research or project on how to do sequencing in the best possible way, please do not hesitate to get in touch with us. We also want our systems to be as flexible as possible so that you can use the same system for multiple applications. Imagine, for example, that you want to deeply characterize a particular cell type, here depicted in the blue cell group, and that you want to see if there's only a single cell type or if it can be further subdivided. You have around 1,000 cells, and you sequence them to quite a high depth of 500,000 reads per cell. So the sequencing output for this study is 500 million total reads. And this is an excellent fit for the SP flow cell. On the other hand, if what you would like to do is more an atlasing approach, you take around 200,000 cells of a particular tissue or of a particular organ to understand what is the cellular composition of that tissue or organ. And you profile those cells at 50,000 reads per cell, needing 10 billion reads in total. And this is an excellent fit for an S4 flow cell. So as you can see, you can do both these studies and also everything in between using the same system, simply by selecting the optimal flow cell to use. So again, we would be more than happy to help you out with any questions or any doubts that you may have about your particular project. Our technologies have been used in a large number of single-cell publications, and here there are simply 4 examples from the different buckets that we mentioned before. You will see an RNA example in the blue area, chromatin accessibility example in teal, proteins in purple, and DNA in red. And if you're interested in reading more, here's a great publication review, which is about over 100-page PDF, showing how single-cell sequencing has been applied across multiple fields, and it also features different methods that can be used, like RNA, epigenomics, and DNA at the single-cell level. And with this, I would like to thank you, and I will pass you over to Stephen Hague from 10x.
Thank you, Aida, for the introduction. So just to run through some information about 10x Genomics to start with. So when we look at the sort of biological system, so we have an example here of looking at the human. So it's an incredibly complicated system to try and understand, and we believe that when we work down to single cell resolution, that we really gain insights to kind of how this organism functions. So we obviously know this, that we have approximately 40 trillion cells in an individual. We have approximately 6 billion DNA bases in a diploid genome, and sort of 50,000 mRNA molecules that can be transcribed. So it's a really complicated system, and really the mission statement of 10x Genomics is to try and make tools that enables the researchers to delve down into that with great resolution and really understand what's going on down at the single cell level. So if we just have a very quick overview of what 10x is all about. So we can see at the top left panel of the slide that we provided a number of instruments which we can use to partition cells into droplets, which you see in the middle portion, and we're able to effectively barcode these cells. So ultimately, when we're looking at gene expression, we're able to do this on a single cell level, and then we're able to build that data back up into fairly complex teasing plots to really understand the biology. I guess where 10x sits as well is that we provide a number of novel assays in biochemistry, and we've invested a lot of R&D effort in analysis software and visualization tools as well. So we really try and support the researchers across the whole area of single-cell genomics. Recently, as you may know, we have also moved into spatial transcriptomics with an acquisition of a Swedish company towards the end of 2018. So that's really going to be my focus on my short introduction today. So 10x has been around since, or released their first commercial product back in 2015, which is really based around link read technologies. But around about 2016 and onwards, we've really focused our efforts into the single cell genomic space. And we've released a number of kits that are developing complexities. We moved from straight single cell gene expression to we're looking at things such as immunology as well, and being able to look at cell surface proteins and look at things like CRISPR profiling as well. And then, as I mentioned, what we've done very recently is we moved slightly left the field, and we've moved into this notion of doing spatial gene expression. So being able to take cryostat sections and really look at profiling what's happening on a molecular or gene expression profile into the individual tissues. So, you know, why is that of interest to researchers? So to move away from the straight single cell genomics field and look more into profiling tissues. And this slide just gives a very kind of quick introduction to the notion of why we might want to do that. So basically in the two panels, A and B, what we're looking at is two tumours. And, you know, visually, when we just examine these, they appear to be very similar on the morphological level. But if we're able to profile these on a molecular level and we're able to look at gene expression, what we can actually see in A versus B, B tumour has got poor prognosis, whereas A tumour has got better prognosis. If we look at gene expression profiles of lymphocytes, then we see actually when we look at this that there's a very big difference between these two tissue sections. So what we're seeing in panel A is that the lymphocytes are actually able to penetrate into that tumour and initiate an immune response and really combat the progression of the tumour, whereas in B, those lymphocytes are held around the periphery of the tumour, so they're not able to kind of effectively fight the tumour. So this is kind of giving us sort of better understanding really into the biology of what's happening here, which we may miss by conventional methods. So that's just really the notion of why people are increasingly interested in the use of spatial transcriptomics. So the solution that we have currently and are also developing in R&D is just shown here. So it's a simple glass slide, and on there we can array capture probes on it. So the slide shown at the top consists of 6 subarrays. So that means in this iteration we can section cryostat section tissue, and then we can mount that onto the subarray. So potentially between 1 and 6 separate cryostat sections on there. And if we look down at the bottom left, when we look at the subarray, then what we're seeing here is in the current iteration, we have 1,007 spots. So we can mount our cryostat section onto the subarray, and we can go through a process of fixing and permeabilizing that tissue. And when we do that, the RNA will diffuse out, and then we can capture that RNA effectively. So if you look over on the right-hand side of the slide, we see the capture probe. And effectively what's going to happen is as the RNA passes down through the cell, that will be captured onto the poly-T capture region. And then what we can do from there is we can amplify that and we can make a library. So what's important when we do this is we incorporate, when we do the first strand cDNA synthesis, we incorporate the spatial barcode into the cDNA, and that ultimately allows us to deconvolute the data back to each one of those subarrays on the slide. And the kind of work that we are seeing or that we will be supporting is, so for example here we can see a section of a mouse brain, and this is looking at the hippocampus, which is outlined in this red region here in the sort of center of the slide, and we can sort of orientate our genes. So obviously what we're not doing here is we're not doing targeted approach to doing this. So this is hypothesis free in a way that we can capture all expressed genes potentially. As shown here, what we can do is we can highlight that region of the brain, and then we can just simply look to see which genes are expressed in there, and we can rank those in terms of the expression profile in there. And moving from there, we can do things as shown here. So we can highlight again hippocampus in a separate area of the mouse brain. And what we can do there is we can compare and contrast the gene expression within the brain there. So you can see down at the bottom right that we can see a volcano plot. So we can see genes that are significantly upregulated, shown on the right-hand side of the slide, and genes that are significantly downregulated, as shown in red, and the ones that remain the same, shown in blue in the central portion there. So we're able to do this kind of compare and contrast between different regions, in this case of the brain, and we're able to really gain insight into the biology of the tissue that we're looking at as well. So that's the current iteration of the spatial transcriptomics, and what we're currently working on is to move away from this a little bit, and we're introducing the Visium process. So this works in a similar way. but we're streamlining the workflow, so that the time to results will be improved or shortened, and we'll also see improved sensitivity around there, and we'll see improved resolution round about this. So this is where we are going with this product, and we will aim to release this in Q4 of 2019. And then just as my final slide, so this is a recent venture that what we're doing is we're actually inviting people to submit scientific abstracts, so 300 words in total, and we will run a Visium project for you. So that's in terms of the sequencing and in terms of the slides, and more details can be found at this 10xgenomics.com challenge spatial. So we invite people that are interested in this and have novel projects. Please feel free to submit an abstract through to us, and we will judge that, and we will choose the winners, and then we will do the process for you. So thank you very much for your time. That's all from me.
Hi everyone, and welcome to this webinar. My name is Thrane, and I'm here today with Dr. Hanna Eriksson to talk to you about spatial transcriptomics and cutaneous malignant melanoma. I'm with the Spatial Transcriptomics Research Group, which is an academic group led by Professor Joakim Lundeberg. He founded the spatial transcriptomics technology together with Assistant Professor Patrik Ståhl and Professor Jonas Rensén, and it was published the first time in Science in 2016. In the lower left-hand corner, you can see a picture of Science for Life Laboratory, or SciLifeLab. This is a research institute in Stockholm where the technology was developed and where the Spatial Transcriptomics Research Group is based. Below the picture, you will find a link to SciLifeLab's webpage, as well as our group's webpage, where you can find out more. I joined the group a little over 5 years ago, and since then I've been involved in various projects. One of them is a collaboration with Dr. Hanna Eriksson, where we use spatial transcriptomics to explore heterogeneity in cutaneous malignant melanoma. And we published a pilot study in Cancer Research last year. Hanna will share some more results from this study, but first I will talk about the concepts of spatial transcriptomics and how the technology has been further developed by 10x Genomics to become Visium. The basic idea of spatial transcriptomics was to bridge histology and RNA sequencing by providing a histology image as well as large-scale gene expression data from a tissue section. Very simplified, the workflow is to stain and image the tissue, construct a sequencing library from mRNA captured from that same tissue section, and then visualize the gene expression with spatial context. This visualization can, for example, be the total gene expression or total number of genes detected, or you might want to see the spatial distribution of one or a few selected genes. This figure shows the location of clusters from melanoma data. Hanna will tell you more about these results later on, and I will also show more examples of data visualizations towards the end of this presentation. In the lower left-hand corner, you see a list of some publications from the Spatial Transcriptomics Research Group. And as you can see, we have applied the method to a variety of organs and diseases, such as mouse olfactory, breast cancer, heart, ALS, and even plant species. You can find more publications on our webpage through the link to the right. The very heart of spatial transcriptomics is the ST slide. It looks like a regular microscopy slide, but has 6 identical capture areas. What I'm showing you here is what we call the 1K slide, meaning that each capture area has 1,000 spots. And this was the first slide version and the one that we used in our melanoma study. I will come back to slide versions in the next slide, but let's first take a closer look at the spots. A spot is a cluster of millions of immobilized oligo-DT primers. These have a spatial barcode that is unique for each spot, and additionally, they all have a cleavage site, a T7 promoter, PCR handle, and a unique molecular identifier, or a UMI. The structure of the primers is a little different in the 10x Genomics Visium, but the concept with the spatial barcode is the same. The spots have a diameter of 100 micrometers and a center-to-center distance of 200 micrometers. Furthest to the right, we can see that one spot is covered by a varying number of cells, and these will all get the same barcode in the sequencing data. Again, the slide I'm showing here has 1,000 spots per capture area. We have since then upgraded our slides to 2,000 spots for more recent projects, and the 10x Visium slides have increased to 5,000 spots per capture area. On the 2K slide, the spot diameter is the same, but they are closer to each other. And on the 10x Visium slide, they are both smaller and denser, providing higher spatial resolution. I will come back to the 10x Visium slide a little later, so stay tuned. Here is a schematic figure of the tissue preparation workflow, that is, all the steps that are carried out on the glass slide. First of all, a tissue section is placed on a capture area, and this is followed by fixation, hematoxylin and eosin staining, and imaging. After imaging, the tissue is permeabilized enzymatically, and this will allow mRNA to be captured onto the barcoded oligo-DT primers. Reverse transcription will extend the barcoded primers with cDNA, and the mRNA cDNA hybrids are then enzymatically cleaved off from the glass slide. We keep the glass slide inserted into a cassette with a rubber mask to get individual reaction chambers for each capture area. Then, after the cDNA has been cleaved off from the glass slide, it will carry a spatial barcode and can therefore be pooled so that we will get one sample per capture area. I will not go into detail on the spatial transcriptomics library preparation, which is quite different from the 10x Visium version, but I thought I'd mention that for the last 2 years, we have been using an automated approach developed by Dr. Anders Jent and published in Scientific Reports in 2016. For the finished library, we use Illumina paired-end sequencing to get the spatial barcode and UMI for read 1 and the gene information for read 2. I mentioned that spatial transcriptomics has been used on a lot of different tissue types, but still the protocol may need to be tweaked a little bit, and especially in the permeabilization step or the permeabilization time. The goal is to open up the cells to maximize mRNA capture, but still keep the structures intact so that the mRNA will not diffuse horizontally and hybridize onto spots further away. For tissue optimizations, we have a separate type of slide that we call the QC slide. However, in the 10x kit, they are called TO slides. Instead of having spots, the capture areas are uniformly coated with oligo-DT primers, and these do not have any spatial barcodes. So here, the mRNA can be captured all over the surface, and then fluorescently labeled nucleotides are added to the RT reaction. We will thereby get fluorescent cDNA wherever mRNA was hybridized, and the tissue is then digested and the slide washed before imaging. The resulting fluorescence image is a cDNA footprint of the tissue, and we can see that it reflects the morphology very well with a strong signal in cell dense areas. However, it's not only about cell density. Some tissue structures or cells tend to be more transcriptionally active. Other than permeabilization time, there are some additional parameters to take into account. For example, we have sometimes had problems with tissue cracking, which is typically a result of the freezing procedure. Sectioning thickness as well as staining protocol may be changed depending on tissue type or preference in a particular study. The RNA quality is also of importance, and we typically extract RNA and check the Win value on a bioanalyzer before we get started with the spatial transcriptomics protocol. I thought I'd mention some of the issues Hanna and I encountered when starting with melanoma lymph node metastasis. The very first thing we noticed was that the biopsies looked very different in size and cell type composition, as you might be able to see from the images. Because of this, we ended up optimizing the permeabilization time for each biopsy, although you would normally only have to optimize once for each tissue type. We also found some biopsies to contain a lot of pigment or melanin. Melanin is known to inhibit reverse transcription, but what we found to be the initial problem was to get these sections off from the glass, which in turn made the optimization tricky because of autofluorescence in the images. In the end, we found a way to get the tissue off, thanks to Janeta Andrusivova, who was working with us for her master thesis. Then we could proceed with optimization, and the results do look promising, but we didn't include these biopsies in our first study. Another thing we were concerned about was the age of the samples. The oldest one had been in the freezer for 20 years when we got started, but that one actually scored a Win value over 7 and gave a decent ST library. So I guess the take-home message here is that although the samples need to be fresh frozen, they do not necessarily need to be fresh. Hanna will continue on the topic of melanoma shortly, but first I will switch focus to 10x Visium, which I got the opportunity to try out in July this year as a part of their beta program. What you see here is a Visium slide and some specifications. As I already mentioned, the capture areas have 5,000 spots, and there are 4 capture areas per slide. The spot diameter has been reduced to 55 micrometers, and the center-to-center distance to 100 micrometers. This means a higher spatial resolution with only 1 to 10 cells per spot, depending on tissue type. Each capture area is marked with a border that is visible in the tissue image, as you will see in the next slide, and this is used to align the sequencing data onto the tissue image. For the beta program, Dr. Anneli Mollbrink and I prepared Visium gene expression libraries from mouse brain. We then sent the sequencing data to 10x for analysis with a pre-launch of their pipeline. Here are some preliminary results that I will go through very brief to give some examples on what you can do with the data. To the left, we have H&E stained tissue with a border around, as I just mentioned. And to the right, we see the total number of unique molecules, or UMIs, detected per spot. And since the scale bar is not very clear, I can point out that all the spots that are bright yellow got around 40,000 UMIs each. Now to the right, we have the total number of genes detected per spot instead of UMIs. And here, bright orange is equivalent to 6,000 genes. As I said in the beginning, we can also focus on one particular gene to see how its expression is spatially distributed. In these 2 images, we have EGR1 and HPCA. And here we see IGFBP6 and TBR1. And lastly, we can cluster the data and then get spots colored based on cluster assignment. And with that, I would like to thank everyone in the Spatial Transcriptomics Research Group, Johan Hansson's group at Karolinska Institute, funders, and of course 10x Genomics.
Hi, I'm Hanna Eriksson, and I'm working as an oncologist at Karolinska University Hospital in Stockholm, Sweden. I share my time between clinical work in the field of melanoma and also clinical and translational research. And today I'm going to present and follow up on the presentation by Kim Thrane, spatially resolved transcriptomics, which enable dissection of genetic heterogeneity in stage 3 cutaneous malignant melanoma. Cutaneous melanoma is one of the most aggressive of all skin cancers, and the incidence is increasing in most countries with fair skin populations. Early detection and surgical removal of the tumor is still crucial for the prognosis and survival in the disease. Early stage melanoma has a relative 5-year survival of almost 100%, whereas metastatic disease used to have a very short survival of 6 to 9 months before the introduction of novel therapies such as targeted therapies and checkpoint inhibitors. The prognosis in stage 3 disease, which is lymph node metastasized melanoma, is very heterogeneous, and the survival varies between 40 to 70%. Melanoma is one of the most genetically but also immunologically heterogeneous of all cancers. Activations of several signaling pathways and of the immune response are involved in tumor progression and therapy resistance, but the biology of melanoma also gives several opportunities for potential novel therapy targets. Thus, further research is needed to elucidate the molecular mechanisms underlying melanoma biology to improve the diagnostics and to identify prognostic as well as treatment predictive biomarkers. The intratumoral molecular heterogeneity in stage 3 melanoma have already been analyzed with an array of analytical approaches, including gene expression analysis, immunohistochemistry, and proteomic approaches. Single-cell RNA sequencing is a powerful tool for addressing transcriptional heterogeneity, but knowledge about the spatial origins of single cells is vital when studying the tumor microenvironment. We therefore aim to optimize and apply the spatial transcriptomics technology for the in situ and quantitative detection of gene expression in stage 3 melanoma lymph node metastasis. And for this purpose, we analyzed the transcriptomes of almost 2,200 tissue domains in fresh frozen tumor sections. By retaining the positional information of the tissue domains, we could superimpose the transcriptomic data onto histological tissue images and visualize gene expression throughout the tumor. So the aim with the project was to optimize and apply the spatial transcriptomics technology for the in situ and quantitative detection of gene expression in stage 3 melanoma lymph node metastasis. And now I will show the results for melanoma. We did a pilot study to assess whether spatial transcriptomics would be useful to study heterogeneity in stage 3 melanoma. First, we optimized the protocol for lymph node metastasis and then prepared the ST libraries on these 4 biopsies in duplicates, as well as had them examined by a pathologist who annotated areas of melanoma, lymphoid, and stromal tissue. The samples had been previously genotyped for some common mutations, and we also had information of the outcome of each patient, and they had been classified as long and short-term survivors. Here is a short data summary. On average, 15,000 genes were detected per sample and around 3,000 unique transcripts per spot. The section-to-section correlation was typically lower in these samples compared to mouse olfactory bulb, likely due to heterogeneous nature of cancerous tissue. Before exploring the spatial data, we merged the counts for each sample to mimic bulk data. A PCA then revealed a strong separation across patients, and we analyzed if ST could detect sub-areas with shared expression profiles. The next results I will show you are based on a data-driven analysis tool that was developed by Dr. Jonas Maskula in the Lundberg group at SciLifeLab. Basically, the data from all spots in all samples were decomposed into 20 factors or clusters, and each cluster was represented by a spatial activity map and a gene expression profile. This illustration is simplified to only 4 factors. The upper panel shows the spatial activity maps, that is the location of a cluster or factor, and below are the top 5 genes for these factors. But actually a set of 50 genes was generated. So now we have these 20 clusters, and I will show a few of them, but here is a first visualization of all factors with T, S, and E in color space. Here, the spots with similar gene expression will get similar colors. Above are the H&E stained tissue images with annotation. And now I will highlight 2 of the samples. Sample 1 and 2 were the only ones that had a lymphoid tissue annotated by the pathologists, and these areas are marked with yellow in the H&E images in the top. In the T, S, and E, they both appeared blue, so it seems that these areas shared expression profiles. The tumor sites, however, appeared to be patient-specific. Also, it is interesting how sample 1 from a long-term survivor had a homogeneous expression pattern in the tumor area, in contrast to the very colorful sample 2 from a short-term survivor. This could represent tumor heterogeneity being related to outcome, but of course we can't draw any conclusion in this exploratory study. When comparing the spatial activity maps to the histological images, we found 4 factors to be particularly interesting. These 2 here both have spatial activity maps that correspond to melanoma areas. So we labeled them melanoma A and B. They overlap to some extent, but not always. Sample 1, for example, seems to have a melanoma A, but not B, whereas sample 2 has a bit of both. The expression profiles of melanoma A and B included both cancer and immune-associated genes. For example, CD63 in melanoma A is reported to act as a tumor suppressor, whereas S100B is sometimes used to follow disease progression. We found one factor corresponding to lymphoid tissue with high expression of, for example, CD74, which has been associated with recurrence-free survival and overall survival in stage 3 melanoma. The lower panel shows a pattern that was not annotated by the pathologist, but with ST. Since it was clearly in between the tumor area and the lymphoid area, we called it transition area. Several of the genes in this factor have been associated with resistance to checkpoint inhibitors. And these genes have also been linked with tumor growth regulation through the GADD45J and K pathway in other cancers, such as in glioblastomas, along with loss of immunogenicity, acquired resistance to PD-1 blockade in patients with melanoma through defects in the pathways involved in the interferon receptor signaling, such as I'm sorry, I had to take it again. Yeah, it was a lot.
No
yes,
problem.
I'm sorry.
No problem at all.
Yeah, good. So I go for this picture again. We found one factor corresponding to lymphoid tissue with high expression of, for example, CD74, which has been associated with recurrence-free and overall survival in stage 3 melanoma. The lower panel shows a pattern that was not annotated by the pathologist, but with ST. Since it was clearly in between the tumor area and the lymphoid area, we called it transition area. Several of the genes in this factor have been associated with resistance to checkpoint inhibitors. and the genes have been linked with tumor growth regulation through the GADD45J and K pathway in other cancers, such as in glioblastomas, along with loss of immunogenicity and acquired resistance to PD-1 blockade in patients with melanoma through defects in pathways involved in the interferon receptor signaling sector, such as B2M and the antigen presentation. We were interested if we could find out more about the transition area, and in order to obtain a more detailed view, we analyzed the samples individually, here with PCA. When clustering the data from sample 1, indeed we found the spots from the transition area formed a cluster of their own. We then looked into what genes that were differentially expressed and had the highest spatial variance and visualized them as heat maps. These are some examples. PMEL and SPP1 are both melanoma markers, and these are clearly enriched in the tumor site. I mentioned that CD74 is associated with better outcome, and here we can see that this gene is highly expressed in the lymphoid tissue furthest away from the tumor, whereas IGLL5, which is also immune-related, is mainly expressed in close proximity to the tumor in the transition area. To summarize, we optimized the spatial transcriptomics protocol for melanoma, and with the data-driven analysis, we found shared expression in healthy tissue, multiple cancer profiles within the tumor, and identified transition areas in tumor microenvironment. The results demonstrate the intra and intertumor gene expression heterogeneity. We generated a more detailed portrait of the heterogeneity among melanoma metastases, and we showed the transition area, which may be a component of to fully understand the tumor progression. In future, integration of the spatial component into analysis of multiple factors underlying tumor progression and therapy outcome would be of interest. And I will thank the patients, the Professor Joachim Lundeberg's group, Professor Johan Hansson's group, the funders, and 10X. Thank you.